Hi, everyone. Welcome to Foresight's Biotech and Health Extension Seminar series sponsored by 100 Plus Capital. Really happy to have so many of you here. Not surprised, given today's guest. Before we hop into today's discussion, I just want to remind people that if you are still interested in joining us for Vision Weekend, which is our annual member gathering, there are still applications open for subsidized attendance. We're going to be in San Francisco. You're going to have the chance to meet lots of people from this biotrack and rejuvenation track, but also across different tracks. We're going to be not only in San Francisco, but also at a castle in France, and there'll be some intermingling between the different venues as well. And so if you want to basically meet top folks in this group, but also across different groups from throughout the year, this is like a really wonderful opportunity to get together in person. Great. I'm going to share more in this chat. There are application-based tickets available. So we have the ability to subsidize a lot of you guys' attendance, and it would be wonderful to see you in person. Cool. So for today, we're so happy to actually re-welcome George Church here. So when I discovered Foresight about like 10 years ago, just before when I joined, I went into our archives and I found, George, you've given a really wonderful presentation to Foresight before, quite a while back. And much of the technology is still really as relevant as when you discussed it back then already. I think apart from that, you really need no further introduction. Everyone here, I think, is well aware of your work, of your countless startups. I don't think I've really met anyone else who spun up so many projects in the biotech and rejuvenation area. I'm going to share much more about you in the chat in case people want to read up the growing list of your endeavors. But for now, I'm like super delighted, honored to have you here. And I'm going to give it up to you now, because I know that we're going to struggle at the end of the seminar in time when people want to get their questions in. So thank you so much for joining. We're thrilled to have you here, and the stage is yours.
Okay, great. Yeah, hopefully there'll be plenty of time for question and answers, because I'm just going to give a representative a couple of examples, and I think some of you know other things that I do that you can ask about. So or about the things I talk about. So I'm going to share my screen. There it is, Foresight. Must be in the right place. Okay. So I work on, I think, a number of things relevant to the Foresight Institute, but I'm going to focus partly at the prompting of the organizers on gene and cell therapies for age-related. We're essentially reversing age-related and infectious diseases. Full conflict of interest in this website down in the lower right. And there we go. So I want to announce in a certain sense a preprint. So this is hot off the presses, where we think we have a way of making any organism resistant to all viruses. We've done it for one organism, I think pretty convincing, including just taking random viruses out of the environment that we know nothing about, thousands of them, and it's resistant, and for good reasons on paper as well. And then that's part of a bigger plan to make enhanced organs that are enhanced in various ways. So not just deal with the organ shortage, which is killing people, but to make those organs as good as they can be, including germline engineering, but germline engineering of animals, not of humans. And then, but in the care of humans. So then all of the above is made possible in multiplex editing, and I'll talk about the dark matter of the genome and how that applies to aging reversal as well. So we are in exponential times, and I think my group has contributed at least as much as any other in both the reading and writing of DNA, which has improved about 20 million fold in cost and a little less than that in quality. And it continues. to go exponential, but many years going faster than Moore's law, 2 to 10 times, 2 to 10-fold improvement per year. And this is, I think, largely due to multiplexing. That would be a whole other different talk. But today, instead, would like to talk about how it is we make decisions for therapeutics or TX for gene therapy, cell therapy, molecular and protein therapies. And my lab and our spinoff companies work on all 4 of those. But in particular, I want to justify gene therapy a little bit. And some of the advantages are on the far left-hand column here. In principle, it can be once in a lifetime rather than taking a pill every day for the rest of your life, or taking 100 pills, worst case, every day for the rest of your life. It's just once and done, in theory, anyway. It is very smart at the delivery system. Almost no other form of drugs is capable of chemotaxis, for example. That's something that's fairly unique to cell therapies. And the mechanism of action that most medical research contributes to is an easy stepping point between that basic research and gene therapy. We typically these days collect information that's formulated in terms of proteins and nucleic and genes behind them. It's very low off target. For example, they have a big multi-gene family that are closely related evolutionarily. It's hard to target, like kinases say. It's hard to target one with a drug, not hit the others. While you can make not only discriminate multi-gene family members, but allele specific, meaning person specific, and splice form specific. So it's very specific, easy to achieve that with gene therapy. And finally, it can be low cost, and I'll mention that in more detail right here on this slide. So gene therapies aimed at rare diseases, including Zolgensma, and the new record holder is $2.8 million a dose for Zolgensma was for spinal muscular atrophy, and the new one is for hemoglobinopathies. Those are cost effective, as high as that price tag is per dose. But I, having helped with the exponentials in reading, writing DNA, would like to see the therapeutics go down in cost. And in fact, if you look at the formulation that is very similar to gene therapy, it's essentially molecularly identical, never called that, is the most recent vaccine round with COVID. All 5 of the top 5 were formulated from gene therapy methods, including viral capsids and double-stranded DNA payload in 3 of them, and messenger RNA and lipid nanoparticles for the other 2. And all of them were inexpensive, the cheapest being about $2 a dose compared to 2.8 million. Okay, so multi-virus resistance. So if the last slide was kind of playing with viruses, both friends and foes, use virus capsid to produce a vaccine that protects you against another virus, AAV against COVID, coronavirus. But here is making multi-virus resistance essentially in one fell swoop by changing the genetic code. So our genetic code consists of 64 triplets of nucleotides that encode 20 amino acids or so. And we've been working on this since around 2004. And here's some of the papers and some of the people that participated in that. But I'm going to jump and I'm going to jump ahead to what we just did in this preprint in bioRxiv. Akush has led this effort, and we have recoded 2 codons, in particular 2 serine codons that encode serine. Serine is normally encoded by 6 codons. In this strain, it's only encoded by 4. and the extra 2 codons are swapped into leucine codons. So these are normal amino acids. We have shown that we can use non-standard amino acids as well, but just for this project, we swapped 2 normal amino acids and produced them at high enough levels that they couldn't be overcome by other transfer RNAs, which is one category of virus that brings its own, some of its own genetic code with it. They don't bring it all with them, and so it's relatively easy to come up with a general hack. Well, relatively easy since 2022. And here's some examples of those where we took mixtures of 12 fresh environmental isolates. These are from farms, and, waste rivers and so forth. And we have done experiments where we have up to 10 to the 12th trillion viral particles per milliliter, and it drops essentially to 0 with and no observed escape so far. And on paper, there's not likely to be any escape because so many positions in the viral genome still depend on those 2 serine codons, and they're swapping in for leucine. So we think this might be it. So, but now let's integrate that as one example of ways of making pathogen resistance and various other enhancements that we would like to the organs that we can produce. We're trying to produce organs both from humans themselves and also through the normal developmental process in a species that looked promising back in the 1960s. That's how long people have been dreaming of using pig organs. But there were at least 42 things that needed to be fixed. And back then, there was no real way of doing that much editing. In fact, even when we started, it wasn't obvious that you could do that much editing in the germline of a mammal and have its, not have lots of problems due to side effects, off-target editing, and so forth. But anyway, this is the wish list from the whole field that had accumulated since the 60s. The top of the list with sugars sounds innocent, but there's three different carbohydrates on the surfaces of all pig cells, and indeed most animals are incompatible as organ donors. And these are very rapid rejections. These are hours, not weeks. And then we get into things like incompatibility of blood cascade, protein clotting, and complement, the major histocompatibility, which is the main thing that we match human to human transplants. And then finally, there was these endogenous retroviruses, which the FDA was not happy that the average pig tissue produces endogenous retroviruses that are where there are 20 to 60 of these viral genomes embedded in the germline of wild and domesticated pigs. And we came up with a way that we could knock them all out at once. And we've done that in multiple strains of pigs. So it's very reproducible. And there's really no excuse to be giving immune suppressed patients organs now that have endogenous retroviruses anymore, because we've shown that they can make it all. These pigs are quite happy with all of these mutation changes, including eliminating all the endogenous retroviruses, which wasn't obvious when we started. And so we have small herds, dozens of these pigs providing organs for transplants in preclinical non-human primate trials. And those are going well. We have 3 clinical efforts. The main one close to us anyway is Jim Martman at the MGH surgery. And we have survival up to 620 days, I think, is the latest. So that's looking very promising for human clinical trials happening soon. These are some of the papers that Luhan Yang, who was a graduate student and a postdoc in my lab and co-founder of two companies, one in China, one in Cambridge, Massachusetts, a real force of nature. And so back to the topic of organ enhancements, which would be very difficult to do anyway, other than germline engineering. But it is inspired by natural and previous experiments, mostly in mice, but now transferred into pigs. They are naturally resistant to many human pathogens, bacterial, fungal, and viral. But in addition, we have the methods that I mentioned before that we'd like to transfer. There are methods known for getting cancer and senescence resistance in mice that we would like to. We would not want our organs to succumb to cancer or senescence if we can avoid it. I've already mentioned immunity enhancements in the previous slide. And then we're interested in cryopreservation, dehydration, DNA damage, for a variety of reasons. One of them is these are related to aging. The other is that they are related to storage of the organs. And here's a couple of organisms that have extreme resistance to tolerance of cryopreservation, including the salamander, which goes for over 45 days at minus 55. So that's, we'd like to get that working for organs in general via germline engineering. We also have a proof of concept. This, like the virus resistance, is done in E. coli, and so it's still a ways to go before we have it working in mammals. But we have, nevertheless, and this is work that inspires us rather than from our lab, we have 10 to the 5th fold improvement in radiation resistance in this normally non-radiation resistant organism with just 4 mutations. And this is very encouraging for things that we're trying to do in parallel on human cells. Now, one of the kind of things that constitutes genome damage, in addition to radiation caused genome damage, is jumping genes, transposable elements. I've already talked about one kind of that, which is the endogenous retrovirus, but even more pervasive example are these long interspersed and short interspersed nuclear elements or jumping genes. And these lines and signs have been kind of a part of the dark matter of the genome that have made it hard to sequence genomes, much less study them genetically. But we think we have a breakthrough on that I'll share with you in just a moment. But first, to just document some of the recent articles, and by no means a comprehensive review of articles that have shown or indicated that these categories of dark matter, repetitive DNA in the genome, are involved in aging, inflammation that's damaging, and related degenerative diseases. And we're tackling essentially every category of repetitive elements, and have recently increased our target from endogenous retroviruses, which were around 20 to 80 copies per genome, to line elements, which are 24,000 copies per genome, to centromeres and sign elements, which are in the millions per genome. And I mean, it seems crazy from our perspective that just a few years ago, it was hard to do one gene at a time, to now be talking about thousands to millions. But I'll show you a little bit more about that. So here's an example where we took human pluripotent stem cells, which are actually from my body, and attack and saturated all of the line elements in that using deaminases, both cytidine C to T and A to G. We far prefer the A to G. It has less off-target, it has less toxicity. I should mention that anything more severe than deamination, where we're just basically changing a nitrogen to an oxygen by in a water-mediated reaction. Anything more than that, like a nick, even a mere nick, much less a double-strand break, at this level of simultaneous editing is immediately deadly. So we have to get everything right so that we have factors that prevent apoptosis and prevent various kinds of repair that can result in double-strand breaks. When we do all of that right, Raphael, Junting, and Corey in particular carrying this on. We have another paper ready to be submitted that follows up on this NER paper. And we have essentially all of the active, the ones that aren't completely crusted over in the genome are, we can saturate, we can get every one of them with this deamination method. Now, putting all that together with what is known about aging, I would maintain that a great deal is known. It's kind of a half empty, half full, but I think we have enough known that we can start turning it into therapies in the same, so much more is known than how much was known about, say, smallpox. When we started doing smallpox vaccines, we didn't know anything about virology or immunology at all. But anyway, we know all of there's 10 major hallmarks and a set of biochemical pathways that go with each of these, mitochondrial, caloric restriction, telomeres, things that you've all heard. These are now all targets for drug development, and I'll give you some, I think, cutting edge things that we're doing. So that 10 goalposts are represented in an interconnected set of reactions, which can be layered on top of the cell, in this case, with the extracellular media on the top of the slide, and then a membrane and transmembrane proteins next level down, the cytoplasm, and the nucleus at the bottom, and mitochondria, endoplasmic reticulum, and other organelles on the side here. And each of these has these enzymes, or the regulatory proteins and RNAs that are well characterized, and many of them are drug targets or are subject to gene therapy. Now, I'm going to pick out 3 of them that we used in multi-gene therapy, because these had the characteristic not of being down here in the genome. We've also done the Yamanaka stem cell factors that you may know about. This is a popular topic because they do literally reverse aging from, say, 80-year-old fibroblasts or blood cells to embryonic. So it's an enormous reversal, but it's hard to deliver. It's possibly hard to keep it in line in the diverse set of tissues it's starting in. But instead, we looked at what was inspired by so-called young blood, where the various factors that have been uncharacterized for most of the years that this has been done. Here we picked 3 that were likely candidates for that sort of phenomenon, which I'll show you. So these are now indicated with these red, blue, and yellow arrows. We have fibroblast growth factor 21, alpha klotho, and TGF-beta receptor. I think the important thing here is these are either soluble or can be made soluble, and we tested them in all possible combinations, as there's been diagrams about, and to see whether any of them interfered with one another, whether they resulted in aging reversal, as assayed on aging diseases, not just biomarkers like DNA methylation or RNA transcriptome, but actually multiple diseases. And I'll show you an example of taken from a paper in PNAS. Noah Davidson was a postdoctoral fellow in my lab and then became co-founder of Rejuvenate Bio. which is applying this both veterinary and human. Started with mice in this paper, and dogs is in clinical trials now for veterinary products because people want a few more years with their pets, but then it's also prelude to human clinical trials because it's looking very good in animals so far. Anyway, so in this first paper addressed 4 different diseases that are age-related. Almost every disease has some age-related component, even falling down and not getting up, and COVID-19 and the cognitive consequences are all very steeply increased with age. But here, the one of the disease models for accelerated aging, or starting with pre-age mice, is we can accelerate the age-related weight increase to the point of morbid obesity, and then restore it with this with these various combinations of color coded on the left here, where it doesn't just keep going to the point of muscle wasting and loss in abnormal anorexia or something like that, but it levels off at a normal level. And then diabetes is another one, disease model where insulin tolerance test, et cetera, can be improved. You can see the same cocktail of drugs that's working for both of these, and is also working here. Not the colors, but the names of the factors for a kidney loss model of unilateral obstruction, and we have another one on heart disease. And Noah Davidson, who was the star of that show, was also involved in a total of 3 out of these 4 papers, all of which used AAV, which is the smallest viral vector for delivery. He was not involved in the cytomegalovirus study, which had one of the biggest viruses for delivery. Even bigger delivery that we do is via cell delivery rather than viral. But in any case, it was basically AAV is about 5 kilobases, cytomegalovirus about 100 kilobases, and the cells can deliver gigabases. So I mentioned the Yamanaka factors here. OSKM, we use 3 of them, OS and K. I mentioned the 3 below, and then telomerase and follistatin were used in the slide. I'll show you in the next slide. And now the list of diseases is 8 different diseases, and keeps growing. Basically, if we get something that one cocktail of drugs that hits all these different pathways, we're probably getting close to the core of aging, and it's easy to get this approved, much easier to get this approved than something that deals with longevity, because our variation in longevity is so great in the population. We would have to do a multi-decade trial to be convincing, and that's too long and too expensive. So even though we do go to the FDA with reversal of specific age-related diseases, if we get any of them, essentially we have an approved drug that could get all of them. But here's one where we did collect some longevity data, even though it wasn't the primary goal. It just fell right out. This is, again, this large cytomegalovirus delivering either telomerase or follistatin genes. And you can see here the wild-type mice control on the far left, dying early, whose death curve in percent probability of death observed, not theoretical. And then 2 different genes, telomerase and follistatin, way far on the right, showing non-overlapping death curves, and inspired by previous literature, not from our life. So I know I went through that pretty quickly, but I know there's also many questions to be asked in the Q&A, and since so many people showed up, thank you. I'm going to open it up for that, but just to quickly review, I shared a preprint where we think we have resistance to all viruses in one cell type, which we are actively now trying to repeat that in pigs and human cells, human stem cells. I described a number of ways we can enhance organs for only for pathogens, immunity, cryopreservation, cancer senescence. And then a lot of this has been helped by multiplex editing, which now is at crazy levels of at least 24,000. And we're working on a repeat family that's in the millions, which can also be leveraged for other things, because it's, for example, recording of information, which we have done as well. And then finally, this aging reversal, which offers a way of getting it among the most general and possibly cheapest category of therapies. It's cheap because the market size is large, like as it is for pandemics. But this might be even larger market size because of everybody is impacted by aging. So we hope to make that accessible to all. So period, full stop. I'll stop sharing. Thank you.
Oh, fantastic. Thank you so much. We're not going to do this thing where everyone unmutes and claps. We have too little time and we need to go through a lot of questions. But this was a really great, well, a few are doing it anyway. But this was really, really great and like such a wonderful, trajectory through the entire field and then like ending on longevity. So thanks a lot. That was really, really useful. We already have one hand raised here by Micah, so I'll take you first and then we move down the list. So raise your hand now or say your question in the chat and we'll get to you as fast as we can.
You briefly mentioned using cell delivery of gene therapies. Can you give like the very short elevator explanation of what that is? I'm familiar with it's like AAV viruses, CMV, and you've got DV dumbbells. I haven't heard of cell delivery though.
Right. Well, that's because it's not as far along. Although the category of cell therapies is often, you can think of it as you can do ex vivo gene therapy on, say, hematopoietic stem cells, reintroduce them into the body, and then they percolate. They contribute to the entire, they go into every part of the body, including chemotaxis. So that's one way. Another way, so that's where the cells are, they're the delivery package themselves and have a large payload of genomic material is edited or engineered. It also, since the viral capsids that we use for gene therapy are made in cells, it is possible, although not yet demonstrated, that you could make them in situ in the body in the right place. So you have the combination of the cells that can chemotax or otherwise find a correct location, plus the viral specificity. That's what I meant by it.
That you just inject into the person, more or less.
You also have the option of injecting it selectively. So if you want to have both anatomical and biophysical targeting, yes. But you could also do it systemically, and in fact, all the gene therapies that I've talked about for aging were systemic delivery through intravenous.
Wonderful. You get a thumbs up. I think question answered. Thank you. Next one we have Reed.
Okay. First, thank you so much, Dr. Church. That was very informative. And I wanted to ask about the question in terms of the gene therapy for intranasal delivery versus injection. Gene therapy for aging, you mentioned briefly in the last slide. What do you think is the main difference and what will be the main barrier for intranasal versus injection for gene therapy delivery?
Well, intranasal has the obvious advantage that you can do it at home. It's not what normally people think about, but it's in principle, you could also do an injection. Most, for example, diabetics do intramuscular injections, on a daily basis of insulin, typically genetically engineered. But I think that's the main difference. It may be particularly appropriate for respiratory diseases, protection against lung cancer and respiratory illnesses of pathogens. But yeah, I think they're not fundamentally different other than that.
Is there any barrier for intranasal delivery though?
Oh yes, I think there's a number of things that would be challenging to do that way, but since cells can, there are cells that can cross the lung epithelium into the endothelium into the blood, and there are other cells that are capable of getting from the intestine across that difficult barrier into the blood, and going then from the blood to difficult additional crossings like blood-brain barrier. In principle, there are a number of interesting options, but for the time being, I think there are things that are more appropriately delivered by, say, intrathecal injection into the cerebral spinal fluid, for example, is a good way of getting into the brain without necessarily having to engineer the crossing the blood-brain barrier. But we have plenty that do. They could preferentially even into the nervous system.
Awesome. Thank you so much.
Yeah.
Wonderful. Next one up with Ageless Partners.
Thank you. And thanks a lot, George Church, for sharing all your ideas here. Just had a question on the one of the slides we were talking about the 4 combination gene therapy. And I was just curious, sort of a multi-part question, but I'm curious, like, are we testing this in humans, and if there's any sort of trade-offs between the delivery mechanisms, like having more than, can you have more than one in this combination? Like if you were doing lipid nanoparticles with an AAV, is there any like complications if you're combining multiple therapies? And I was also curious, out of that 4 bunch, if there was like one in particular for osteoarthritis, or if it was a synergistic thing between more than one.
Right. So we did find one indication, one disease where all 3 together. So in general, all 3 together is a good combination. But we found some indication for where we drop one of them out. My guess is that at some point we will add some others in, because we really want to get all 10 pathways of aging at once. Because if you leave one opening, that's what will kill you. In fact, it's been estimated that if you got rid of one of them, you might add 2 years to your life. But we want to get rid of all of them. So we haven't got the final list of genes yet, but those 3 that Noah Davidson used look particularly promising, and including osteoarthritis.
Nice. And is it currently being tested in humans already?
It is. We have not started human clinical trials yet. We've started FDA-approved dog trials, which is very close in many ways in size and in environment and food and so forth. But they, as soon as those are completed or nearly completed, we will start the human clinical trials, which could be 2 years from now.
Gotcha. Thanks. Appreciate it.
Yeah.
Wonderful. Larry, you're up next.
Yeah, hi, George. Interesting stuff. I mean, I was curious, about your codon change thing. AG, I think, the AG serine codons, I think they are used in a lot of human proteins. So I didn't quite understand how it would work, to actually get, viral inhibition exclusively. And then the other thing on your adenosine doesn't mutate directly to guanosine. It goes through inosine, and there is an inosine repair mechanism, but you're saying that's deficient in cells, or you think adenosine deaminase is a target for aging? I mean, I think it's an interesting thing. I'm just not that familiar with what you with those things. Yeah.
Right. Two good questions. It is true that both the human genome and the bacterial genome that we did these experiments in, you have 20,000 examples of those codons being used in the protein sphere. But what we did is we mutated all of those places so that they're no longer used. We swapped them out into the synonymous codons. So there's 6 different codons, and we eliminated 2 of them by mapping them onto the other 4. So in those cells, they're only using 4 of the 6 codons. So that's null for a while. And then we put in a new tRNA that turns those codons from serine to null to leucine. So we kind of go through a null intermediate. But yes, the host has been heavily
protecting E. coli anyway from phages. So yeah. But
what's that?
Yeah.
I'm sorry, I missed that.
Yeah, we're protecting E. coli from phages.
That's correct.
Yeah,
that's correct. That so far it's just been done in E. coli, but it's been done by a method that we think is completely general, which is changing the genetic code. And we are partway into doing it in human stem cells. And we will do plants and domesticated animals as well. Anything that's got a serious viral problem of economic significance. But we don't have that yet. Anyway, so to answer your question, we did the genetic engineering so that the host is no longer dependent on those 2 codons. And your second question was about adenine is indeed goes through inosine on its way to guanine. It's a simple, the repair is typically done by replication, and the replication of the I typically takes it to a G. And if it doesn't, then you can do another round of mutagenesis for the same deaminase. So,
but there's a repair mechanism for that too, George, right? I mean, the repair does recognize inosine and gets in there, and yeah.
It is not nearly as efficient as the repair mechanism for cytosine.
Okay. Yeah.
And in fact, so
it's another aging clock potentially. Then, if you take a cell that hasn't replicated and a cell that replicates a lot. Yeah.
I don't. That wasn't part of our plan. We'll take it under advisement. The plan was just use adenosine deaminase as a simple way of doing editing that's non-toxic. It is the least toxic way of doing multiplex editing we found by far.
Okay.
Wonderful. Great. Next up, we have Krian.
Thanks, Allison. Hi, George. It's been a while. I have a sort of vague question that could go different ways, but it's this. So if you have this scheme whereby you can, if I understand it correctly, make cells resistant to viral manipulation, how do you route around that if you want to use viruses to manipulate cells? That's the first thing. And then this related thing is, I know in another part of your world, you have, at least in the past, done these sort of broad mass sequencing gathers in the sea and all kinds of places, and presumably in our guts, you name it, right? So what I'm wondering there, and this is how it relates, is if you could say something about the genetic diversity of the microbiome that we carry around, including the virome that we carry around. How many viruses are in us, and what are they doing? And if you shut off this viral stuff, as you're alluding to, does this have sort of broader species or ecosystem consequences? Like, are these viruses just working against us, or is there something deeper going on. So first question, simple. How do you route around,
Yeah.
can you do viral vectors if you've turned off viral infection? And secondly, what about the broader virome and what's going on there?
Yeah. Well, the first one is slightly easier. Both of them are great questions. If we want to use viruses, as I mentioned, we used viral delivery in 3 out of the top 5 COVID vaccines. In that case, it's typically a synthetic. We completely get rid of the viral nucleic acid. We're just using the viral capsid. We still, some kind of nucleic acid makes that capsid, but it can make it in a completely different cell, a non-recoded cell. Then the payload itself is synthetic, and it can be whatever you want. So you can use the new code and would use the new code for that, whether it's messenger RNA-based vaccine or viral DNA based. So that's all synthetic where we can and do handle them. But these are not typically full viruses. These are hybrids of viral capsid proteins and a payload, which is non-viral. And then to the environmental components, we have found some bacteria that are helpful that are in our gut. Not typically pathogens, just the opposite. They're helpful bacteria. So we would want to eliminate those. We have not yet found a viral component that I'm aware of, other than, ironically, the endogenous retroviruses that we eliminated in pigs. But even there, we showed that what we did was we knew that one gene of the virus was helpful, which was the envelope gene, but its ability to hop around and to cross species was not necessary for that. The envelope gene is involved in making the extraembryonic tissue, so it's involved in the maternal fetal communication in the placental. So we did not eliminate that. We simply eliminated the polymerase gene, which is required for its hopping around, which is probably detrimental. We'll keep our eyes open for other viruses in the environment that might be helpful, but I think many of those functions, if they ever are found, could be substituted by either a recoded version of the genes or some other mechanism. But like I say, there's none that we know of yet. But if anybody finds one, I'm all ears.
Great. We have a few more questions here next. Let's do one by John Ferber, and then I will take a few final questions.
Hi, George. It's been a while since before the COVID pandemic. Some types of gene therapy only require a few cells or a few types of cells to be altered. I wanted to alter every cell in the body. Is there a technology that would guarantee getting it into every cell, including past the blood-brain barrier?
Almost. So I left out a few slides that cover that, but we have a couple of my companies are working on targeted delivery, in particular therapeutics. We published a Nature Biotech paper where we showed that we could engineer it and select for various tissue deliveries, including brain delivery, and but many others. We could do these simultaneously we design a library of 1 million. This is not random, but machine learning based design, and that million then sorts itself out in various tissues in the body, and then we look for which ones are enriched in the different tissues. And we have found that using machine learning, we can up the amount of change that we can tolerate from, with naive non-machine learning based methods. It's hard to get beyond 4 out of 28 positions. Let's say we take a region of the genome, 28 amino acids long. If we tried to get 4 out of 28, we're getting low single-digit % survival. And if we go above that, we get undetectable survival. But now with machine learning, we could get up to 28 out of 28 amino acid changes, which is quite high.
Well, related to that, I'm very interested in doing something about line one, messing up the genome. And I'm guessing that that's important in every cell in the body and that there's a lot of line ones in every cell. And I'm wondering how we can possibly interfere with, delete, or somehow prevent damage by line one in most or all cells.
Right. So unlike the endogenous retroviruses, the lines are, a very, very distant cousin. There's no known positive effect of lines. It looks like there's some negative effects. And in the same way, we can knock out the endogenous retroviruses by hitting the reverse transcriptase gene. We can hit the analogous conserved active site in line elements. And so we're doing that now in animals to see whether they could truly survive multi-generations without active line transposition or reverse transcriptase. That we don't know yet. It's my guess that will be a net positive, but we have now the genetic tools to ask that question.
Thank you. And the final, have you published on it?
We've published the line element mutation in human cells. We're just starting the animal experiments.
Wonderful. Thanks. In the last 5 minutes, I would love to get to the questions that we always ask in the session, which are, number one, for someone new entering your field. And I mean, your field is so broad. Let's keep it perhaps on the, aging, longevity relevant bits of the field, which is of most interest to people in this group, if you may. What would someone be working on? If they're coming into the field, like what would make your work in that, as long as as much as it relates to longevity and aging, like much easier? Like what's an undervalued challenge where you're like, hey, if you're new to this area and want to take something on and make my work much easier, this is what you do.
Yeah, I think multiplex editing, where you can make a large number of edits, is very valuable. It's challenging to say do genome recoding, which requires so much multiplex editing. I think that's a bottleneck. Most groups, including most pharmaceutical companies, rarely go beyond 1 or 2 genes at a time. We're going into the tens of thousands. So that's one thing I think is quite valuable. I think that using bio delivery is still not a solved problem. I said that we had ways that we can really get high levels of mutagenesis and delivery to any target, but it's still it's a far cry from getting delivery to every cell or even every stem cell in the body at 100%. Now, fortunately, you don't need 100% delivery, but I still think there's quite a bit of room for improvements. So multiplex editing and delivery mechanisms.
No, okay. And then if we, let's say, make much progress on this, could you, kind of like leave us with perhaps like a vision of like what could be possible in a, case where everything goes right in longevity, based on the tools and mechanisms that you're developing in like 5 to 10 years?
Well, I won't abuse the everything goes right card. Try to keep it realistic but yeah, I think it's quite possible that we could make a low cost gene therapy, $2 a dose that is once and done, that hits a large fraction, let's say 90+% of the key cells, not necessarily every cell, and results in a rejuvenative process that is somewhat similar to what we've observed with the cocktail of young blood factors and the Yamanaka factors. So some set of results like analogous to what we've seen, but in a clinical setting applicable to almost every cell at high efficiency. And that could have implications for diseases, not just but diseases of poverty, because it is affects so many diseases, and it is it's going to be low cost. And then other tools, maybe not directly aimed at aging, could further impact diseases, infectious disease and disease of poverty, and that would have a positive feedback loop of economic significance worldwide.
Well, it's a nice existential vision of what's possible here. And then, really finally, this is like a shameless plug moment. What is there anything that people can concretely do to really advance your work? So usually, like, if people are currently raising funds for like another startup, or if they have like a new paper out that they want people to read, this is a moment where is there anything that this group, which really appreciates your work, could do to advance your individual work?
Well, I think it could be a mutual benefit, and I kind of shamelessly plugged all the way through this talk. I'm not sure we need a separate plug, but yeah, we're a very collaborative group, and happy to work with people, both started companies, which is the way we get it into the real world and out of the purely academic ivory tower, to better ways of doing clinical trials. That's something that's quite challenging, but you can see progress all the time. It's the veterinary trials go about 5 times faster. The COVID-19 clinical trials set a record at about 1 year instead of the usual 10. So I think there's a lot of potential there as well.
It certainly is. Hey, thank you so much. This was absolutely wonderful. And yeah, you've had a really full house. Thank you so much for joining us. It was really a pleasure to have you on. I hope it wasn't the last time. If you do have, future updates on your work, like this group is certainly pretty interested, as you can tell from attendance. Thanks a lot. And thanks everyone for joining. Just a reminder, I put a nomination form here in the chat. If you want to nominate future presentations, it's here in this chat. Thank you, George.
Okay.
Let you get on.
Thank you.
And thanks a lot.
Take care.
Bye, everyone.
Yeah.
See you on
Bye.
the next one.
Yeah. Bye.